I think the announcement of the meeting has misled the perception that I was just awarded the prize, and so I got many congratulations over the day, and thank you so much for being 15 years late on this one. So, but it's a really great opportunity to really come back and talk a little bit about what has developed from actually we got awarded for as a team. As I said 15 years ago, and I just want to give you a short overview of where our research interest has developed over that period of time. So what I'm clinically interested in as a clinically trained and active endocrinologist is generally very prevalent diseases, which is diabetes on one hand and obesity on the other hand. And so we're really facing an obesity epidemic, and we know that the increase in obesity prevalence, having approximately more than 30% of the population being obese, predisposes for a whole plethora of diseases, but maybe most importantly, or among many others, for type 2 diabetes, which in parallel keeps rising as obesity goes up. And so what really has been fascinating me over the last 3 decades is a very fundamental question: how is body weight regulated? I mean, how can we manage, in light of thousands of calories that we are facing every day, manage to take the right decision, if everything goes well, to exactly eat as much as is needed to maintain a stable body weight? And so what really has hindered the advances in the field of obesity research for a long period of time was that we did not have a molecular correlate for the regulation of body weight homeostasis. And so it was Kennedy who, in the early 50s, proposed a hormonal feedback regulatory principle of body weight regulation, where he predicted that signals would exist which originate from the periphery of the organism, which would be secreted in proportion to energy availability. And he proposed the existence of receptors for those signals in the central nervous system, which then would adapt food intake and energy in accordance to the energy state of the peripheral organism. And again, this was in the 50s, and it was a beautiful model, but it lacked any molecular correlate for the following 40 years. And it was really the breakthrough discovery of the identification of the hormone leptin by Jeff Friedman's group at the Rockefeller University, who really revolutionized our understanding of body weight regulation. And so Jeff had spent more than 10 years of his scientific endeavor to try to understand what is the genetic defect underlying this mouse mutant. And this was a spontaneous autosomal recessive mouse mutant called the obese mouse, OB mouse. And so these are siblings. And you see that the mouse carrying the mutation has about double the body weight, if not even more, than the normal controls. And so then he spent, at the time, between the 80s and 90s, about 10 years to positionally clone the underlying gene defect in this mouse mutant and identified the gene mutation which made this mouse obese. And he termed the gene which carried the mutation leptin, originating from leptos in Greek for lean. So it was the lean signal that was mutated in this mouse. And it turned out that leptin fulfilled all the prerequisites and the predictions that Kennedy had made 40 years ago. So leptin is exclusively produced in white adipose tissue in proportion to fat storage. So the more energy we have, the more leptin is produced and released. And the leptin receptor exactly resides in the key neuronal centers in control of food intake and energy expenditure, particularly in the hypothalamus. And now if you take recombinant leptin, you inject it into the brain of a mouse or a rat, it potently suppresses feeding, and it activates energy expenditure, really closing that gap on the homeostatic feedback regulation of body weight, food intake, and energy expenditure. And so what we have been focusing on over the last 20 years, and the whole area of research has focused on, was try to understand, now knowing what the signal is that comes from the periphery, we can now ask the question, which specific neurons in the brain specifically respond to leptin, and how is the biological action of leptin then translated through activation of the brain? And I would like to introduce to you a very basic neurocircuitry which resides in a very old region of the hypothalamus, the so-called arcuate nucleus of the hypothalamus. And there are about 3,000 cells which are characterized by the expression of a certain neuropeptide, pro-opiomelanocortin, which is cleaved to alpha-MSH. And those POMC expressing neurons are activated by leptin when energy stores are full. Alpha-MSH is released, and that potently suppresses feeding. So really, this is why those cells are called anorexigenic food intake suppressing neurons. And right next to those cells, another 3,000 to 5,000 cells sit, which look alike under the microscope, but they do the exact opposite because they express different neuropeptides. Those cells are inhibited by leptin, so they are turned inactive when energy stores are full. They're activated during starvation when leptin levels drop, and when activated, those cells potently promote feeding. So you really have this brake and gas pedal on the regulation of food intake and energy expenditure. And so we really wanted to study the functional role of those two neuronal populations by now 20 years ago. And this was work by Eva Gropp, a clinician scientist in the group at the time, and she devised a mouse model where we could inducibly and selectively ablate those 3,000 POMC neurons or the 3,000 AGRP neurons in adult mice by diphtheria toxin-mediated cell ablation. And if you remove those 3,000 AGRP neurons, which are activated during starvation, the animals rapidly lose weight and suppress feeding, while removing the neighboring 3,000 cells leads to a steady increase in food intake and body weight. So really showing you that we have this very fundamental system of a relatively sizable number of neurons which are in critical control in the regulation of food intake. And what really was the philosophy, and I'm not going to go deep into this for the sake of time, but what we reasoned was then if those cells are specialized to sense the energy state of the organism by integrating the signal such as leptin, and we showed that they also respond to insulin, which is a mediator of how much glucose is available in the organism. Those cells can directly sense fatty acids, they can sense ghrelin, which is released from the stomach upon starvation. So those cells are really optimized to integrate all signals about the energy state of the organism. And so we then reasoned, well, wouldn't it make sense if they not only control how much energy we take up from outside the body, but also how we partition energy within the organism. And through a series of studies, we could then demonstrate that the same neurons not only control feeding and energy expenditure, but also hepatic glucose production, lipolysis in white adipose tissue, and more unrelated biological functions related to the energy state, such as the control of innate immune responses, bone mineral density, and through ascending projections to the brain to adapt also mood and behavioral responses. And so I just want to show you one example of a very recent study where we have shown that those AGRP neurons, which are activated during starvation, have a strong regulatory effect in liver. And on a pathway which is very fundamental to protein quality control and energy regulation, which is autophagy. So autophagy is a pathway which is usually considered to be activated in a cell-autonomous manner upon energy deprivation. So if a liver cell is deprived of energy, AMP levels goes up and AMP kinase is activated in a liver cell, and this will stimulate the recycling of lipids and proteins in the cell to provide energy sources. But what we could demonstrate is, you see when you fast an animal, this is the increase in autophagic flux in liver, but if we artificially block the activation of those hunger-activated neurons, this completely abrogates the ability of the liver to induce autophagy in response to short-term starvation. So really telling you that there are different phases that sure, during prolonged energy deprivation, the liver is capable of cell-autonomously activating this pathway, but there's even an earlier response when those specialized neurons start sensing the upcoming energy deficit, that they already prime the peripheral function to adapt to that change. And so really the brain instructs peripheral tissues to prime for responses which are anticipated to be needed for full physiological adaptation. I would like to switch to an aspect of those neurons which really has been keeping us busy for the last couple of years. So if the model that I told you was exclusively true, that POMC neurons get activated in response to leptin exclusively, you would have a clear prediction. You have, in a starved state, POMC neurons would be inactive. You eat something, calories are absorbed, they're stored in adipose tissue, leptin levels go up, and then the neurons would start firing. Now what we can do using modern molecular systems neuroscience tools is that we can monitor in freely behaving mice the activity of molecularly defined neurons. And this is what Klaus Brandt, a postdoc in the group, did. So he put a calcium sensor, a calcium-sensitive fluorophore, specifically in POMC neurons, and then we can implant a fiber and we can record the activity of the neuron while the animal is running around and freely behaving. And so what you see here is calcium recording from that food intake suppressing POMC neuron. And the moment you drop in food, the neurons start firing, which is completely inconsistent with the model of hormonal feedback regulation, right? So basically, the moment the animal sees and smells the food, the neuron starts behaving as we would have predicted it to occur after the calories have been consumed. And just to show you that this is really dependent sheerly on the sensory food perception, we presented the animal with something that we called caged food. So we took a food pellet, wrapped it up in a wire mesh, put it in front of the animal so the animal can see and smell the food but not consume it, and the POMC neuron starts equally responding. So this really tells us that energy state regulating neurons are already activated upon the sheer perception of calories that will be presented to the animal. So if this signal if then the calories don't come, this signal tempers off relatively quickly, while when the animals then really eat, the second and third phases of that regulation kick in. And this is just summarized here in a recent review from us in the scheme, that in addition to this long-term hormonal feedback regulation, you have this very acute sensory food perception dependent regulation of those critical metabolism regulatory neurons. And so the question really was, what is this signal good for? And so this is just showing you the summary of the study, which we published some years ago. And what Klaus could demonstrate is actually that if the animal only sees and smells the food, POMC neurons get activated. Within a minute, you get the sympathetic nerve activity input to the liver, which initiates a very coordinate response, which is characterized as a classical ER stress response. And this is a pathway that has been beautifully worked out by Peter Walter and colleagues, which was always assumed to operate in a cell-autonomous manner. So the ER stress response is usually activated when unfolded proteins accumulate in the ER, initiating a transcriptional response to counteract and to increase protein folding capacity. And so basically what Klaus could demonstrate is that the neurons hijack that cell-autonomous pathway to prime the liver for the anticipated need to increase protein folding capacity, protein secretion in the postprandial state. And when Klaus did some experiments to look really at the morphology of the ER, so you can see that actually the ER expands morphologically when the animal just sees and smells the food, we also saw that there were morphological changes in mitochondria occurring at the same time. And so this is work on a follow-up project from Sinikka Henschke, who was a PhD student. And so Sinikka really wanted to see, are there rapid changes in liver mitochondria upon the sheer sensory perception of food, even before the calories are being consumed and metabolized. And so what Sinikka did is, she performed a phosphoproteomic screen in liver. She took fasted animals and either exposed them to caged food, so food that was not accessible, could only be seen or smelled, or did a real refeeding experiment. And then she isolated mitochondria, performed a phosphoproteomic screen, and really identified specific phosphosites which were gradually increased upon the sheer sensory perception of food as well as upon refeeding. And so when we zoomed in which proteins exhibit that pattern, we were really struck by a certain protein, which is a mitochondrial fission factor, MFF, which got specifically phosphorylated on the residue serine 131 when the animal was just seeing and smelling the food. Since we had previously shown that the sensory food perception dependent regulation of liver depends on POMC neuron activation, she performed a complementary screen where she optogenetically activated those 3,000 cells. So there was no food involved, the animal was starving. We just turned on the activity of 3,000 neurons, performed a phosphoproteomic screen, and she identified the exact same phosphosite being activated when just those 3,000 neurons are activated. So what is MFF? So mitochondria constantly undergo fusion and fission events, and MFF is a critical adapter protein which sits on the outer mitochondrial membrane and recruits a protein DRP1, which then wraps around the mitochondrion to execute fission and fragmentation of mitochondria. And so the first question was, what is the kinase responsible for the phosphorylation of MFF serine 131? So this serine sits in a perfect consensus motif for an AKT kinase, which is a proteotypic insulin signaling mediator. We then performed in vitro kinase assays showing that recombinant bacterially purified MFF can be phosphorylated by AKT, which is inhibited by an AKT inhibitor. So then we generated two complementary mouse models where via CRISPR-Cas9 gene editing, we mutated that serine either to a non-phosphorylatable mutant or to a phosphomimetic mutant, and then could use cell models and those mice in vivo to study the functional consequence of this serine 131 phosphorylation. Since AKT is proteotypically activated by insulin signaling, we could show that in wild-type cells, these are mitochondria, so you really have this interconnected mitochondrial network in the basal state. If you incubate the cells for a few minutes with insulin, you really see those punctated mitochondria. In other words, fission takes place, the mitochondria start fragmenting. And if we do the same experiment in the non-phosphorylatable mutant, insulin fails to induce mitochondrial fragmentation, and this is all quantified here. And so the big question is now, what does this translate to in vivo? And this actually was a very surprising finding. When we then looked in vivo at the mice which no longer could phosphorylate that residue, which no longer could induce mitochondrial fragmentation in response to insulin, those animals were insulin resistant in vivo. We can confirm this in a pretty sophisticated experiment, which is a euglycemic hyperinsulinemic clamp, which I'm not going to go into great detail. But basically what we find is that the insulin resistance is mediated by a failure of insulin to suppress hepatic glucose production. And this is what we're really excited about, because this identifies a completely non-canonical pathway of how the acute change in mitochondrial dynamics is linked to insulin's ability to regulate hepatic glucose production, and the dysregulation of which, of course, is a key event in the development of type 2 diabetes. So for the last 2 to 5 minutes, 5? Okay. I just want to expand the view a little bit, because you think that we're really stupid just working for years on 3,000 neurons on one and the other side. And there are, of course, many different cell types in the hypothalamus. And so what really did strike me, there's more and more data coming out on single cell and single nucleus sequencing data. But one of the problems is there is about 20 studies, or had been 20 studies out there for the hypothalamus. And each and every research group then clusters the cells in accordance to their own studies. And so there's really not a unified nomenclature of the molecular characterization of those neurons. And instead of doing more and more sequencing, we had really ingenious bioinformatics PhD student, Lukas Steuernagel, joining the group. And he took on the task to try to find the optimized way to integrate all the different single-cell, single-nuclear sequencing data from the hypothalamus to basically correct for the batch effects, to maintain the biological truth and difference, to provide a unified neuron hypothalamus single-cell sequencing atlas. And Lucas really did a great job in doing this. And he succeeded in integrating all previously published single-cell sequencing data, combining it with some few single-nucleus sequencing data. And this just gives you an overview that within the hypothalamus, the key regulatory center of energy and glucose metabolism, we can identify at least 250 molecularly defined neuronal cell types. And so I've been only talking about 2 of those, AGRP and POMC neurons. And we really feel that there is room for a lot of discovery for other neuronal types to be identified with critical functions in metabolic control. And I want to give you just a very last example for one of those. Because when we think about obesity development and disease progression, what we usually tend to do is we try to understand the physiology, we try to understand POMC, AGRP neurons, and we assume that they are dysregulated in obesity, which is the case. But by only zooming in onto the cell types which we know already about, we might be missing cells which maybe are only activated under the disease state. And this is a project that Alexander Jais took on. He was a postdoc in the group. And he performed an unbiased screen to identify neurons which are specifically activated when animals are just exposed for a short time to a high-fat diet. And he employed an approach which is called phosphoribotrap profiling. So almost all signals that activate a neuron will activate mTOR signaling. mTOR signaling activates the phosphorylation of S6, which is a ribosomal protein. So if a given neuronal cell type is overactivated in one compared to the other state, in that cell type, more S6 will be phosphorylated. You can precipitate the ribosomes, compare then the mRNA expression between the two conditions, and you will enrich the molecular signature of neurons which were activated in one state. And this is exactly how it worked for Alex. He identified a completely new population of cells in the arcuate nucleus which are selectively activated under the disease condition after 3 days of high-fat diet feeding. And those are characterized by the expression of a different neuropeptide, pre-pronociceptin. And just to cut a long story short, what Alex can show is if he artificially optogenetically activates those cells, the animals start eating more. So the cells which are activated in high-fat diet feeding can promote overeating. And if we selectively ablate those cells, here you see the localization in the arcuate nucleus, you see the successful ablation. This does not change body weight when the animals are fed a healthy, normal chow diet, but it largely attenuates the obesity-inducing effect of the high-fat diet. So really pointing to the identification of a different type of neuron, which seems not to be that important for the regular regulation of feeding, but which seems to gain more importance when we're exposed to calorie-dense, high-fat, high-caloric diet. So really to sum up the talk, I hope I've convinced you that the melanocortin, which is the joint nomenclature for those AGRP POMC neurons, coordinate the adaptation of integrative physiology to the energy state of the organism, and this is really what fascinates me. It's not only about feeding, it's not only about glucose metabolism, it's really trying to adapt whole body physiology in accordance to the energy state. In addition to homeostatic hormonal feedback regulation, they integrate already the sensory information to prime anticipatory physiological responses. Sensory food perception controls liver proteostasis, ER stress, autophagy via melanocortin neurons. Sensory food perception rapidly modulates mitochondrial dynamics in liver, and this dynamic stimulus-specific MFF phosphorylation is required for metabolic homeostasis and the adaptation of hepatic glucose production. And we really think that those PNOC-ARC neurons provide an attractive target for novel obesity treatments. And I think I've acknowledged the people who've done the work along the talk, a lot of collaborators and the funding sources. And again, also my thanks to the Jung-Stiftung which 15 years ago really gave an important contribution for us to pursue that research line. And thank you very much for your attention.